In this video we will cover how to correctly gauge your samples in the NMR's spinner turbines and load the sample changer. When you enter the NMR lab you will see at the computer workstation the user log book. You should begin by entering your name, group, the date and time you are starting. Across the room you will find the station for properly inserting your samples. The station has spinners which each sample must be inserted into, some chem wipes and isopropanol for cleaning your tubes and the depth gauge. Here is an up-close look at the gauge. The gauge has a window where you can see the tube when it is inserted. There are two lines that mark the top and bottom of the NMR probe coil. This is the region of the sample that the instrument detects signals. With samples of 40 mm or greater the solution should fully fill the window. If your sample is much less than 40 mm you should align the sample to fit within the two lines as symmetrically as possible. Samples that are a little taller but less than 40 mm, and the length extends outside the top and bottom lines, should be placed symmetric about the center dots of the gauge. Next, it is time to insert the sample into the spinner and properly gauge the sample. Take a chem wipe and apply a small amount of isopropanol and wipe the outside of your NMR tube to assure it is clean. Pick up a spinner. Make sure you only hold it by the top part that has the stripes. Slowly push the tube into the top hole of the spinner. Make sure the tube remains aligned with the spinner or you could break the tube. The O-ring in the spinners can be pretty tight, so proceed with caution. Push about 4 to 5 cm of the tube past the end of the spinner and then place the spinner into the gauge. Slowly push the tube down until it touches the bottom of the gauge. If your sample is 40 mm or higher, the sample should be properly gauged. If your sample is shorter, adjust it in the gauge as shown earlier. Next, take your sample to the sample changer that is mounted on the magnet. The sample changer on the 300 has a carousel that holds up to 24 samples. Each holder is numbered 1 through 24 and the carousel rotates clockwise. The holder position directly under the vertical column was the position that the previous sample was run from. The next sample position is the holder directly to the left of the column. Note that the red bar before the column is a safety switch that prevents tubes that are too tall from breaking as the carousel is rotated. Be careful that you do not bump into the bar because it will trigger a fault and keep the system from proceeding. The status light in the upper right will turn red. If this happens at any time, please see the staff to clear the issue. Carefully place your sample with the spinner into the carousel. Note that you will have to place it with a slight angle to avoid the safety bar. When your experiments are complete you can remove the sample from the carousel. Note that if you ran just one sample, the returning position is right under the column. Be very careful removing the sample by tipping it on an angle to avoid the column. Tall samples might have to be pushed slightly into the spinner to gain clearance. Return the spinner to the counter with the gauge. This concludes this video on sample gauging and loading. Thanks for watching.